Hey everybody, I just finished Collagen Mutations Result in Osteogenesis Imperfecta from Khan Academy's Biological and Biochemical Foundations of Living Systems passages, and I did pretty well. I thought this was a really short passage. As I approached the first question in this passage, where does the mutation that causes osteogenesis imperfecta first occur? I went back to the second paragraph immediately. I saw them start to define osteogenesis imperfecta, which told me, they didn't really talk about it in the first paragraph. Read through it really quick, and immediately I thought this was almost a joke. It said typically results from genetic mutations in collagen genes. So one of the answers was collagen gene. Clicked it, and sure enough, that answer was correct. All right, so second question. If collagen were treated with protease X, which cleaves at the carboxyl terminal of glycine residues, and protease Y, which cleaves at the amino terminal of proline, then what would be the length of the majority of resultant fragments? This was kind of a trick question, but when I found in the first paragraph, they were talking about the structure of collagen, and they said collagen is distinctive in that its subunits are composed of a regular arrangement of glycine connected to proline connected to X, where X is any amino acid. Here they said protease X cleaves at the carboxyl terminal, the C terminus of glycine residues. So if we were looking at this from glycine to proline to X, it would cleave at the end of glycine, and protease Y, which cleaves at the amino terminal of proline, which would be the beginning of proline. So they'd be cleaving in the same place and leaving fragments of 3 residues in length, the same as it would be right there. I think they were kind of trying to do that as a trick, but it didn't work. All right, number 3, purified collagen protein samples were collected from the tissues of 4 different subjects. Which cell isolate would correlate with a patient with osteogenesis imperfecta type 1 and a patient with osteogenesis imperfecta type 2? In the second paragraph, they talked about here, the most common form of osteogenesis imperfecta, type 1, arises from a dominant mutation, which is a little odd. Well, maybe not odd, but odd for me because we typically talk about recessive mutations. It arises from a dominant mutation in the gene, which results in a deficiency in collagen production. This is important because they then say types 2, 3, and 4 are caused by dominant mutations that alter the structure of collagen. So if you were looking for the difference in a graph between who has type 1 and type 2, type 1, you're going to see less total. It's going to inhibit the production of collagen, cause a deficiency. So on this graph, the only one that stands out is patient B. And when you look down at the answer choices, patient B is the only one for osteogenesis imperfecta type 2. And then they said patient C for osteogenesis imperfecta type 1. And then next to it is just patient C for type 2, which makes sense, I guess. It could be any of the types, I suppose, because I don't know anything about it. Okay, next question, question 4. If a patient comes in with osteogenesis imperfecta type 1, which of the following statements is most likely true? So type 1, we read, was a dominant mutation. That's a deficiency in collagen production, and that's all they really told us. So when we look at the answers, I see one of the alleles has a silent mutation. That doesn't really, maybe, I don't know, but that doesn't make sense. One of the parents has osteogenesis imperfecta type 1. That would make sense, because if this was a dominant mutation, then that would mean that one of the parents had to be a carrier, right? And if the child inherited this, they picked up that allele from the parent, then that meant that the parent had that allele, and if it's dominant, then that parent had osteogenesis imperfecta type 1. It's possible for the child to not inherit that, but if they do inherit that one allele, that 50-50 chance from mom or dad, it's not 50-50, but crossing over, people, learn it. But if they do inherit that, then they will have osteogenesis imperfecta type 1 as well. So the answer here was one of the parents has it. Okay, question number 5. If one allele of the whatever gene was mutated so that the new motif was TRP PROX, so they replaced a glycine with a tryptophan. This is where knowing your protein biochemistry really helps. You replace a glycine with a tryptophan. Tryptophan's very big, glycine's very small. Um, what disease type would this patient most likely display? So when you look down, no disease will be displayed, type 4, type 1, or this mutation will be lethal. So no disease and lethal are kind of ruled out because they didn't talk about lethality in any of this. And then no disease will be displayed. It doesn't seem like it because they're talking about a disease. So they did say in the second paragraph that types 2, 3, and 4 alter the structure of type 1 collagen. So if you replace glycine with tryptophan, there's no reason for us to believe that it would either be benign or it would be lethal. So based on what the passage gives us, we have to assume there would be some kind of problem. And if there was some kind of problem between type 1, which causes an actual deficiency in production, and type 4, which causes just an alteration of the structure, we've kind of already said it already that this is just going to alter the structure. Glycine's very small, tryptophan's really big. It's going to throw off the structure of collagen a little bit, possibly in a bad way. Probably in a bad way, because why wouldn't it be like that in the first place?